DNA Depot presents How Are Bacteria Identified? Kit DD240. Performing this experiment will allow you to stain bacteria and yeast and to determine their size and shape. You will be using specific stains that are used for bacterial staining. This experiment contains reagents and disposables for 6 experiments. Your kit should include 2 microorganisms, Bacillus species and yeast, crystal violet gram stain, iodine substitute, bacterial growth medium, microscope slides, plastic troughs, sterile transfer pipettes, and microtip transfer pipettes. You will also need a microscope that can magnify 10 times to 100 times, a microwave, immersion oil, paper towels, disposable laboratory gloves, and goggles. Preparation of a disinfectant. Make sure a container with disinfectant is readily available for students to use. We recommend a 10% solution of bleach. Using the experiment tube, mix 450 milliliters of water and 50 milliliters of bleach. At the completion of the experiment, soak all plastic pipettes and slides in a disposable container for 4 hours in a 10% bleach solution. Recover plasticware and slides and discard in trash. Flush the liquid in the toilet. Activity 1. Preparation of bacterial cultures. The teacher will assign or demonstrate to the students. Bacterial cultures should be prepared 1 to 2 days before Activity 2 is to be performed. Tube number 1 has a white filter paper that contains dry frozen bacteria. The bacteria will grow and convert the clear medium into a turbid solution. Tube number 1 has Bacillus, which is gram-positive bacteria. Tube number 2, yeast. 1. Carefully remove the cap from tube number 1 and add 2 milliliters of bacterial growth medium using a sterile transfer pipette. Two, cap the tube. 3. Gently swirl the filter paper to make sure that the bacteria is introduced into the medium. 4. Repeat steps 1 through 3 with tube number 2. 5. Incubate tube number 1 at 35 to 37 degrees Celsius for 12 to 16 hours or overnight, or at room temperature for 48 hours. Incubate tube number 2 at 30 degrees Celsius for 12 to 16 hours or overnight, or at room temperature for 48 hours. 6. Do not uncap the tubes. Check the bacterial growth by examining the turbidity of the medium. The cultures need to be turbid to proceed to Activity 2. Caution: turbidity is due to bacterial growth. Gloves need to be worn. The area of the experiment should be wiped down with 70 to 95% rubbing alcohol, and hands need to be washed after the experiment. Activity 2. Preparing slides for gram staining. Make sure that a container with disinfectant is readily available for students to use. We recommend a 10% solution of bleach. Using the experiment tube, mix 450 milliliters of water and 50 milliliters of bleach. Wear gloves and make sure that you read the laboratory safety section on page 3. 1. Using a waterproof pen, label 2 slides, number 1 and number 2. 2. On the bottom of the slide, use a wax pencil or permanent marker to draw a circle the size of a dime in the middle of the slide. 3. Open the tube that is labeled number 1. Use a sterile swab to remove a small amount of the liquid culture. Recap the tube. 4. Place the wet swab on the top of the slide within the circle to make a bacterial smear on the slide. take the used swab and soak in the 10% bleach solution. 5. Let the slides air dry completely. 6. Repeat steps 2 through 5 with tube number 2. 7. After the slides are visibly dry, place them on a paper towel and in a microwave-safe plastic container. 8. Microwave the slides for 5 to 10 seconds to fix the sample and make it ready for staining. Activity 3. Gram staining of bacteria. Caution: when handling bacteria, gloves need to be worn. The area of the experiment should be wiped down with 95% rubbing alcohol before and after the experiment. Wash hands thoroughly after the experiment. 1. Place fixed slides on the plastic trough. 2. Using a microtip transfer pipette, place 3 to 4 drops of Gram Crystal Violet to cover the smears on each slide. Time for 30 seconds. 3. Rinse the slide with slow-running tap water, or use a water bottle or pipette to rinse the slide over the trough. 4. Place 3 to 4 drops of iodine substitute on the smears. 30 seconds. 5. Rinse the slide with slow-running tap water, or use a water bottle or pipette to rinse the slide over the trough. 6. Gently blot water from the slide with a paper towel. Do not rub. 7. These slides can be stored in a drawer at room temperature. Do not stack them on top of each other. 8. Discard contents of the trough in the sink and run water for 2 to 3 minutes. If performing this experiment at home, you can place paper towels in the trough to soak up the contents of the trough and discard it into the trash. Bacteria are all around us. Some are helpful, while others can be harmful to humans. Gram staining bacteria allows healthcare professionals to identify microorganisms and to select the correct antibiotics to use for treatment. Bacteria that stain blue are gram-positive and have different cell walls than bacteria that stain pink that are referred to as gram-negative.